Hi, welcome to the Accelerated Biosciences presentation. My name is Yuta Lee. I am the founder and CEO of Accelerated Biosciences. Today we'll be talking about the ultimate breakthrough in human trophoblast stem cells, or HTSCs. Accelerated Bio is sourcing the highest quality immune privileged HTSCs with no socio-ethical controversy. We're a private company. The technology was discovered in 2003 by Professor Yuta Lee, and the company was founded in California in 2013. Our primary focus is to develop and supply well-characterized cell types from its primary HTSC source. And to date, we've built a robust IP estate of more than 28 patents. A bit about myself: I have more than 10 years experience in cell therapy development and more than 17 years of experience in medical devices. I did my undergrad at UC Berkeley and received my EMBA at China Europe International Business School in Shanghai, China. The inventor, Professor Yuta Lee, is a physician researcher. He's a KOL for OBGYN in Taiwan, got his MB in Kaohsiung Medical University, MD at Tohoku University in Sendai, Japan, and PhD at St. Bart's of University of London. We're working with Martina Molsbergen of C14 Consulting. She comes to us with more than 25 years of business development, marketing, and entrepreneurial experience in biotherapeutics, immunotherapies, and regenerative medicine. She has an impressive track record and closed over $2.5 billion in deal value. She got his Bachelor of Science in Chemistry from Drexel University. Regenerative medicine is the future of medicine. Gene and cell therapy promises to deliver cures that have only been palliative through drugs till today. The regenerative medicine market was reported to be $13.3 billion in 2019 and expected to grow by 23.8% CAGR to 38.7% by 2024. The main challenge for regenerative medicine today is sourcing the ideal stem cell. You're always choosing trade-offs between ethics, scale, and immunogenicity. At the bottom here, you'll see a timeline of fetal development where most people get stem cells. Human trophoblast stem cells are sourced between 4 to 8 weeks post-fertilization in the embryonic stage from an ectopic pregnancy. These cells are mortal and they are pluripotent. HTSCs are unique and an ethical stem cell source. On the left side of the slide, you'll see a picture of a blastocyst at 5 days post-fertilization. At this stage, the blastocyst is differentiated into two types of cells: the inner cell mass on the inside, which develops into the fetus, and the ring on the outside, which is a trophectoderm, which later develops into a placenta. Now, using a human embryonic stem cell from the inner cell mass destroys the blastocyst and is considered unethical. Our cells, HTSCs, are sourced from ectopic pregnancies. And to the right of this slide, you'll see a picture of an ectopic pregnancy. What happens is the blastocyst is traveling through the fallopian tube and it gets stuck. And it continues to grow until it gets too large and it bursts the tube. The mother may experience extreme pain, thinking it's an appendicitis. She'll go to the ER, get scanned, and the doctor will tell her that she has an ectopic pregnancy. And they will then schedule an emergency salpingectomy procedure where they will go in and take out the abortus. How we get the cells is we put a stop to the standard of care, and we go in and we extract the chorionic villi, which is the most distal end from a non-viable fetus. And we give the abortus back to the surgeon. The surgeon then gives it back to the pathologist, who then confirms that it is an ectopic pregnancy and is thrown away as bio-waste. HSCs is a superior stem cell source. Here we see a table comparing all of the stem cell sources. The first two are human embryonic stem cells, HESCs, and induced pluripotent stem cells. These are gene-modified adult cells published by Shinya Yamanaka back in 2006. HSCs are the earliest of the mortal cells that we find. They have no immunogenicity, and the reason is because the baby, developing fetus, the DNA is a combination of the father and the mother's DNA. So why is it that it's able to attach itself to the mother when it's supposed to be recognized as foreign? It turns out that the cells of the fetus, developing fetus, express a surface marker called HLA-G, and that prevents that from happening. Our HTS cells have this expression. We provide speed and scale. The cells have a 22-hour population doubling time, and we've been able to consistently get it over 75 population doublings. So this is a table that shows you the population doubling potential and the doubling time. And with 100 million cells per dose, we're able to create 1 to 10 to the 28th amount of doses. This is pretty significant amount of scale. HSCs are the holy grail of stem cells. We're able to differentiate into all the cells in the body: mesoderm, endoderm, and ectoderm. So far, we've been working with differentiation to NK cells, cartilage, bone, and fat. These are mesenchymal. And then neurons for ectoderm, pancreas and liver for endoderm. We've also evaluated their secretome, which might have some interesting uses going forward. And we are currently developing a HSC-derived IPS line. We've demonstrated immune privilege over 5 animal studies in immune-competent rats, 2 in Parkinson's models, 1 for traumatic brain injury, these are for neurons, and 1 for type 1 diabetes, and 1 for acute liver failure. All of the experiments were done with immune-competent rats. We find that the cells home and regenerate the affected organ. We found no teratoma or tumor formation, and we also see behavioral improvements. We sent out the cells to IDEX Bioanalytics for the H-Impact-1 and to test for 25 human pathogens, and they all come back completely negative. So these cells are pathogen-free. We're working with Wilson Sonsini for over 10 years now, and we have gained 28 patents, and we're working with 7 patent families, and we still have many patents in application. So HSCs has a powerful list of advantages. They're pluripotent, they're scalable and fast, they're ethically sourced, they're immune privileged, they're pathogen-free, they have a chromosomal stability, and an encumbrance-free IP estate. We formed a partnership with IO Biosciences based out of Philadelphia. They have an amazing technology called GEMS, which effectively creates a parking lot in the genome and it effectively eliminates off-target insertions and random insertions. And so what we did was we created a GEMS HTSC, and we loaded a car, and we set it off to kill a tumor. And in our proof of concept, our HSC GEMS was able to eliminate tumor cells. So we used CD19 as a proof of concept, and with the target only on the left side, and with the HTSC-derived NK cells, we were able to eliminate the tumors by 36.7%. And when we fit the CD19 CAR in the GEMS, we were able to almost double the amount of reduction. Our business model is partnering and licensing, so please contact us if you are interested in using HTSCs as a platform for your cell therapy. Thank you.